Hello everybody, I am Hari. Welcome to my class. So in today's class we are going to learn something about biochemical evolution. So what is biochemical evolution, which mainly includes the origin of first life form. What are the sequential actions that have happened for the life to get originated. So that is nothing but the biochemical evolution. So let's try to understand that. So the earth is originated almost 4.5 billion years ago. And the life is originated almost 3.5 billion years ago. Even still we can see certain modern bacteria that resemble the characters of the bacteria from prebiotic world. So what is meant by prebiotic? Pre means it is before, biotic means life. So before the life is originated, whatever the environment is there, so that is called the prebiotic world. So here we can even consider the organisms which were formed in the earlier stages. So those organisms also can be considered under prebiotic world. So even still we can see the modern bacteria that carry the characters of bacteria from the prebiotic world. So what might be the reason? There must be something that is making them to carry the characters from prebiotic world to the current generation. So it must be the evolutionary path. There must be some evolutionary path to carry those characters. So since we humans were not there at that particular time to understand how the life has originated, we must construct some hypothesis. So that hypothesis is nothing but the hypothetical evolutionary path. So to understand those conditions which promote the life to get originated, we are constructing a hypothesis. So that is nothing but the hypothetical evolutionary path. So hypothesis means it is nothing but the assumption. Okay. So this hypothetical evolutionary path must contain certain stages. So the stage 1, which comprises the generation of key components. So why we are starting it with the generation of key components? Why not directly with the origin of life? So as we have already discussed, the earth is originated almost 4.5 billion years ago, and then the life is originated almost 3.5 billion years ago. So it took almost 1 billion years of time for the life to get originated. So what might be the reason why the starting life form has taken that long time to get originated? So here it is very clear that the conditions for those billion years of time, the conditions were not favorable for the life to get originated. So once the conditions were favorable, then it started to get originated. So it's important for us to first understand the conditions of those prebiotic world that promoted the life to get originated. So the conditions of the prebiotic world. So the prebiotic world, we have been studying since so many years that the conditions of the prebiotic world or the atmosphere of the prebiotic world was very much reduced. So the reduced environment here, the reduced environment, so all the compounds that were there in that particular environment, they were in the vapor state. Okay, and the oxygen content was almost very less, and the temperature, extreme temperatures, since the conditions were reduced, all the gases were in the vaporized state. That is because of the extreme temperatures. So the reduced environment and the extreme temperature promoted or made the conditions to be favorable for the life to get originated. So let us discuss about those conditions. Many scientists have come up with many experiments to prove or to make us understand about the conditions of the prebiotic world. But among them, one experiment was successful to explain about the conditions of the prebiotic world. So that experiment was given by two scientists, Miller and Urey, during 1950s. During 1950s, these two scientists have set up an experiment to explain about the conditions of the prebiotic world. So for this, they started mimicking the conditions of the prebiotic world by taking certain compounds. So here they have taken methane, ammonia, hydrogen and water. So by taking all these compounds, they gave the conditions like electric discharge and stimulating lightning. So these conditions were given to mimic the environment of the prebiotic world. So they have taken the gases, they have taken certain compounds and they gave extreme temperatures in the form of electric discharge and the lightning. So then they started getting vaporized. After that, by the condensation process, they got the mixture of certain compounds. Mixture of specific or non-random compounds. So why we are saying specific or non-random compounds means those compounds were specific components, just if they were not formed just like that. Every time they performed the experiment, they got the same result, same compounds. So those were the specific or non-random compounds. So these non-random compounds were nothing but the key components. So these were the key components for the life. So these were the key components for the life to get originated. They are nothing but biomolecules. So we all know what are biomolecules. Biomolecules include amino acids, proteins, carbohydrates, lipids and nucleic acid bases. So at the end of the experiment, they got the mixture of specific compounds. Those are nothing but the biomolecules. So let us know about what are those specific compounds that they got at the end of the experiment. So those specific compounds or biomolecules that they got were amino acids like glycine and alanine. Here they got around less than 2% of the yield, glycine and alanine, and also they got complex amino acids like glutamic acid and leucine. Of course the fraction was very small, but still they got the specific compounds like glutamic acid and leucine. And they also got certain carbohydrates. So they got different kinds of sugars. So among them ribose is the most important one, because ribose is one of the main components of nucleic acids. So they got the ribose sugar, and this ribose sugar has come from the condensation of formaldehyde. So we may get the doubt where from the formaldehyde has come. So as we have already discussed, whatever the compounds were taken, once they got vaporized, they will be leading to formation of different compounds, and those compounds after condensation will be developed into the specific compounds. So from the formaldehyde after condensation, ribose sugar has formed, and then they got nucleotide bases like adenine. So adenine has come from hydrogen cyanide. So after hydrogen cyanide is condensed, adenine base is formed. So even hydrogen cyanide is one of the most likely component of the reduced environment or the prebiotic world. And they also got certain compounds of lipids. So this way they got the specific compounds at the end of the experiment. So which is nothing but those specific compounds, key components were formed in the prebiotic world. So we may get the question that if only specific compounds were formed, what about the remaining compounds? Like here they got very few amino acids. What about the remaining amino acids? Just they got one or two bases. What about the remaining nitrogenous remaining nucleotide bases? Here we must understand one thing, that whatever happened in the prebiotic world was completely uncertain. So despite those uncertainties, the compounds that were formed, again they were combined with one another in a particular assortment in different fashions, and then they have started forming the remaining compounds also. Okay, so here we got very few amino acids, but by different assortments among the prebiotic molecules, even the remaining compounds also they have started forming. So we all know that the carbohydrates generally exist in two forms, L configuration and D configuration. But here in living organisms, generally D configuration is more prominent. It might be because even though the two configurations were there, the compounds with two configurations were there, but D configuration might not be chosen because it might be more suitable, for the physiological activities that have to be happen in the living organisms once the life is originated. So this way, even though different combinations of the compounds, prebiotic compounds, were formed in different fashions, the ones which are needed for the physiological activities might not be selected by the world, by the prebiotic world. So here we are saying the compounds, all the biomolecules whichever are needed for the life to get originated, they were formed. But all these specific compounds were formed by non-biological processes. So what is non-biological process? We all know that in the prebiotic world, till then there was no life form. So whatever the compounds that were available in the surrounding environment, they were combined with one another when the conditions became favorable, and then they started forming the specific compounds. So this way the biomolecules were formed just by using the compounds in the environment, but not by the living cell. So since they were formed without using any life form, so they were formed by non-biological process. Okay. So in the first stage, all the key components that were needed for the life to get originated were synthesized, and then they might have undergone the replication or the reproduction process. So it is nothing but the multiplication process. So once the compounds were formed, they have to be multiplied. If they were not multiplied, just that small quantity of the compounds will not be enough for the life to get originated. So before the lifespan gets over, each and every compound that was formed, they have to be multiplied. So the compound has formed and it has to be replicated or multiplied. Again this has to be multiplied. Again this. So whatever the compounds that were formed, they have to be multiplied until they increase their concentration. And this whole thing has to be happen before the lifespan gets over. So before the lifespan of every compound gets over, the compound has to be multiplied till it reaches the certain limit. So even we can observe this for living organisms. Once the living organism is formed, before the lifespan of the organism gets over, it will be replicated or reproduced. So to increase its concentration in the given genetic pool. So the same way here, the molecules that were formed, once they were formed, they have to be multiplied. So now the key components are produced and they have increased their concentration. So now everything that is needed for the life to get originated was readily available in the environment, and also the conditions were favorable. Now by interactions among the biomolecules that were available, they might have started forming the initial life form. So if there were no interactions, we cannot imagine the life to get originated. So we know we have different interactions like van der Waals forces, hydrophobic interactions, hydrogen bonding, covalent bonding, ionic bonding. So we have different types of interactions. So by using those interactions, the biomolecules, they have started interacting with one another and they might have started forming the living cell. So the first thing, it might be with the formation of the genetic material, and it might have started forming a membrane around it. So membrane, nuclear membrane. So this nuclear membrane is also a type of plasma membrane. So once the nuclear membrane is formed around the once the nuclear membrane is formed around the genetic material, all the components which are needed for the cell, they might have accumulated around this nuclear membrane, and then the formation of plasma membrane might have occurred. So we all know what is plasma membrane. Plasma membrane is nothing but the phospholipid bilayer with hydrophilic heads towards the outer side and hydrophobic tails which are embedded inside because they are hydrophobic. So the lipid molecules, phosphate, and also we can see intrinsic proteins, extrinsic proteins, cholesterol, certain sugars. So all these biomolecules might have interacted and they might have started forming the membrane, that is nothing but the plasma membrane. So if you observe the cell structure keenly, it will be definitely nothing but the biomolecules with specific interactions, right? So this way, the initial cell might got originated by specific interactions among the biomolecules that were readily available in the prebiotic world. So now, the unicellular life form is originated. So now the unicellular life form has originated, and then it might have established the mechanisms for the life to get sustained. So once the life is originated, it has to be survived, right? So for that, it might have started certain biochemical process. So now the life form is there, and the chemical energy was readily available in the surrounding environment, and solar energy was also available. Using chemical energy and the solar energy, it might have started forming the compounds that were needed for the physiological activities. Once these compounds were formed, they might have adopted certain physiological pathways to make the simpler compounds or simpler molecules. So simpler molecules will be synthesized, and once they are used up, they will be degraded by the degrading mechanisms. Again, if they are required, they will be synthesized by synthetic mechanisms. So by adopting different pathways, the cells, unicellular organisms might have evolved into different groups. A single life cell has started forming, and this life cell, depending on the adoption of different pathways, it might have been evolved into different kinds of unicellular organisms. So 1 way, 2, 3, 4. So depending on the variations among them, it might have started forming into different kinds of unicellular organisms. And then the evolution mechanisms might have allowed them to get differentiated depending on the conditions that they were facing. So if you see the unicellular forms, living cells, so these unicellular living cells, they will be grouped into different colonies. So here all the colonies, or simply we can say colony 1, colony 2, 3, 4. So depending on the variations among them, or depending on the conditions that the cells can sustain, the cells were divided into different colonies or groups. So these are nothing but the clusters. So we can simply say cells with a similar nature might have grouped together and they might have started forming tissues, and those tissues with the specific function might have grouped into organs. So from the organs, it might have led to the formation of multicellular organisms. So from the unicellular organisms, by allowing different evolutionary mechanisms, it might have led to the formation of multicellular organisms. Once the multicellular organisms are formed, they have to undergo replication or reproduction process to increase their species in the given genetic pool, and also they evolution and also variation is one of the most important factor for the evolution. Variation means there is nothing but the change. At least if one new character is not included into the given genetic pool, we cannot expect all these different kinds of multicellular organisms, right? So for evolution process to be continued, always variation is the most important factor. So by carrying certain different characters, different kinds of multicellular organisms might have started forming. One which can show the resistance will be naturally selected by the natural selection mechanism, and it will be continued for the next generation. So this way evolution process will be continued. Hope you all understand. Thank you.